single-celled green algae, and there's an ice log in the back which receives light and converts light into electrical signals through proteins that convert light into a flux of protons, calcium, sodium, and potassium. So when the light hits these, the ions flow through, and the algae then modulate how they swim around, which is how they move in a body of water to photosynthesize more optimally. Now it turns out that we're very lucky. This molecule was encoded by a small gene, just several hundred bases of DNA long. And so we could take that gene, which we got from some colleagues, and put it into a virus, and deliver the virus into the brains, even of living mammals. So this uses technology trends from other fields, another theme, I think, of the burgeoning field of neurotechnology, gene therapy in this case, which were developed for other purposes, but which are very powerful in the context of facilitating the usage of neural tools in intact systems. So then serendipity part 2 kicked in. These were genes from algae and bacteria and other small critters. They didn't have to work. But it turned out that these molecules had the right safety profile and speed and magnitude of effect to express really well in brain cells. And so we worked pretty much on the first trial. We found that these molecules would express really well on the surface of cells in the brain, which is where the voltage is created. And when you shine light on these cells, it was enough to activate them and make them do what you want. So the net result is there's all sorts of things you can do in terms of applying these tools to activate sets of cells and to shut down sets of cells. And I think several of the other talks you'll hear about will talk about the applications of these tools, so I think I will skip over that in the interest of time, except to really point out the idea here that a lot of what we do in neurotechnology is about trying to optimize our serendipity. It's very hard to plan on a new tool, but by connecting the dots, or finding something in the natural world, or synthesizing ideas from the old literature and the new, or rebooting an old field that had encountered failure, and now the world's different because we have computers or some other context change that makes things possible. We can actually start to build technologies at an increasingly rapid pace. So flashing forward to the current day, we've now found many classes of these so-called optogenetic molecules, opto for light and genetic because, again, we lucked out and they're encoded by very small genes. And we can put them into cells in the brain and turn on or off these cells, which gives us a lot of insight into the causal consequences of a pattern of brain activity in a disease state or in a disease remedying state.